Biology Part 18. Simple nucleotide substances, ATP and NAD. Nucleotide substances have many vital functions in the cell in addition to their role as subunits for nucleic acids. Adenosine triphosphate provides an important example. As illustrated, ATP consists of molecule of adenosine linked to a chain of 3 phosphate groups. ATP is an effective phosphorylating agent. Under suitable conditions, the terminal phosphate group can be transferred to a molecule of different substances. This increases the reactivity of the second substance and allows useful work to be done. The overall reaction involves the hydrolysis of ATP as summarized in the equation. Note that energy is available as a result of the transfer of phosphate group. The transfer of the second phosphate also yields 31 kJ per mole. On the other hand, hydrolysis of the covalent bond linking the first phosphate to the ribose sugar provides a maximum of 12 kJ per mole. Within the cell, the breakdown of the ATP is coupled to other reactions which require energy. As a result, much of the energy released is immediately transferred into new chemical bonds, with only minimum amount being allowed to escape as heat. Virtually all the complex biological compounds found in the cells are built up using energy obtained from ATP. In order to renew their supplies of ATP, cells carry out the chemical process of respiration. In the laboratory, energy can be released from carbohydrates such as glucose simply by burning it in air or oxygen. However, in the living cells, breakdown of the glucose is a stepwise process during which the glucose molecules is progressively dismantled. Building up nucleic acids. Two types of linkage between the individual and nucleotides are involved in building up nucleotides molecules of nucleic acids. In the first type of linkage, a connection is formed between the sugar group of one of the nucleotide and the phosphate group of the next. The phosphate backbone is a sugar that structures of the DNA and RNA both involve chains joined in this way. There are two important constraints in the way in which base pairing can occur. The first of these is that the sugar phosphate backbones of the two strands must run in opposite direction. This allows projecting the nucleotides to be fitted together. The second constraint is that the base transpairing normally occurs only between particular pairs of the base. In DNA, the possible pairs are adenine, thymine, AT, guanine, cysteine, while RNA are the pairs of adenine, uracil. The guanine cytosine in molecules of DNA, adenine and thymine are linked to hydrogen bonds. Consequently, it is impossible for pairing to take place between A and G or between C and T. As you can see it in the figure, each pair, one has basic belongs to the double-ranged purine family, while the other belongs to a single-ranged pyrimidine family. It also allows the overall length of all the base plane pairs that is the same as the link stands of the held constant distant apart. The individual hydrogen bonds are weak, but provided the sequence of the base on the two strands of complementary, that is provided by the strands of fit together correctly. They are extremely stable. Three-dimensional structures can result. The most important of these are called the double helix.